Well, thank you very much, Fernando, for the introduction. I would also like to thank Fernando and Steve for giving me the opportunity to come up this afternoon and present some of our work on whole body approaches and some of our thinking, our systems approach and thinking in this area. So as Fernando said, I'm a professor in across engineering and medicine at the University of Melbourne. And I'm going to begin with a bit of an overview of some of our thinking, and then I'll move on to a specific study where we've looked at whole body approaches, specifically in the context of aging, in the context of brain and body aging. And please feel free to stop me if you have any questions. Don't hesitate to interject. More than happy to respond. So I'm a neuroimager. I'm a brain imaging person. And as the brain imagers in the audience would know, we use modalities, not only PET, but also MRI modalities, functional diffusion and structural MRI. But as neuroimagers, we invariably study the brain in isolation of the rest of the body. But we know that the brain is intricately connected to virtually every other organ system in the body. And this is not only via the nervous system, but also via hormonal networks and circulatory system and so on and so forth. And as neuroimagers, we ignore this, right? We cut the head, we cut the brain off at the neck, and we only focus on what occurs within the skull. And what I hopefully want to convince you today of is the importance of understanding and integrating the function of other organ systems into our study of the brain. Now, of course, this is not a new concept. It's important to acknowledge that over the years there has been attention drawn to particular axes such as the brain, heart, brain, gut, and perhaps some of the less well-known axes such as the liver, lung, and kidneys. And there is a wealth of work that has been done in this area. What I see as the next step, though, is to integrate across these axes to really start to develop and establish a whole of body approach. That said, it's also important to recognize some of the work that has been done to investigate the impacts of other organ systems on brain function and structure. And this is some quite old work now from Katie Chang and colleagues, which looked at the heart and lungs as confounds in brain imaging. What I'm showing you here is a well-known brain network referred to as a default mode network. It's the regions there shown in red. And what Katie shows here is that the default mode can be localized with more precision using fMRI techniques if we account for the effects of respiration and heart rate as a confound. The point I want to get across here, though, is that we need to go beyond thinking these respiratory and heart signals as confounds, but actually modeling them in and taking account for their effects. So a couple of years ago, Ivanhoe and colleagues proposed the concept of network physiology, which is really the first step, a systems-based approach to understanding interactions across organ systems. And so network physiology, as I've quoted here, network physiology focuses on dynamical interactions among organ systems based on continuous streams of synchronous recordings. Recordings of key physiologic parameters and output signals from multiple systems. And there are various ways to conceptualize network physiology. The first is horizontal integration. This is where we're looking at interactions across different organ systems. And this is typically done at the macroscopic scale. But it's also important to understand that this can be done at different spatial and temporal resolution. So rather than looking at the scale of a whole organ, we can go down to particular tissue types, to cells, subcellular. And what I haven't shown here on the slide is even to go down to genetics and gene expression data. And so we refer to that as vertical integration. There are, however, huge challenges in doing that in terms of integrating data across organ systems. And some of these have already been touched this afternoon. First of all, whole body imaging is costly and it's technologically demanding. Secondly, integrating data, particularly across different imaging modalities, is challenging. And we need to develop new methods, new statistical methods to understand and to integrate data across modalities. And the third point here is, I think, one that's often overlooked, but it's a really important point, is that we're drowning in data. With multi-organ imaging, we've got significantly greater volumes of data than with unimodal approaches. And there are challenges in terms of not only storing this data, but also in computationally processing and integrating data across organ systems and across modalities. So now I want to focus on some work that we've done recently on bringing together not only imaging, but other phenotypes, other blood chemistry, and related phenotypes to understand or to elucidate a multi-organ perspective of aging. I think many of you would have heard of the concept of brain age. There's been a lot of work that's been done on this. The basic idea is that we can take your brain scan, feed that into a machine learning algorithm, and it will predict an age for you. And that age might be either larger or smaller than your chronological age. And so the idea is, if it's larger, that indicates that your brain appears older than a person of the same age and sex. Now, it's been shown, as I've shown on the right here, that brain age is associated with certain demographics and environments. So, for example, musicians generally show a lower brain age, so their brain appears younger than the general population, matched in terms of age and sex. But there are certain diseases and characteristics, such as whether you're born before or after the Dutch famine. This was a study that was performed in the Netherlands, that dictate whether you have an older or younger appearing brain. There's also many studies that have looked at brain age across neurological and psychiatric disorders and found profound differences in brain age. So as I said, the brain age gap concept it's a really simple concept. We can train a machine learning algorithm on a large normative population. We give the machine learning algorithm a person's MRI scan. That could be either structural, functional, diffusion. We tell the machine learning algorithm that person's chronological age, and it learns to predict the person's age based on the MRI data that we propose, that we provide. We can then give it a new person's MRI data and say, well, what's that new person's age? And as I've said, in some people, it will predict the age is greater than their chronological age. In other words, the gap is positive. Their brain appears older than peers of the same age and sex. In contrast, it may be that the gap is negative. In other words, that person's brain appears younger. There are, however, certain caveats that need to be considered here. I'm not going to go through these in detail, but an age gap doesn't necessarily mean that person's brain is aging faster or accelerating. For example, the gap might have emerged very early on in development and remain constant. So we still detect the gap when that person's 40, 50, or 60, but the gap might have come about during development or adolescence. That's an important caveat to take into account. So in this work, what we aim to do is to extend the concept of brain age to other organ systems. And this would give us a so-called clock, a biological clock for each of many organ systems. And this then enables us to probe whether there is synchrony among the clocks. So, for example, if a person's heart appears older than peers of the same age, does this mean that their brain or other organ systems would also show aging? Moreover, are there influences? If my brain is aging faster, does that necessarily mean that my heart or kidneys are aging? Are there causal influences across organ systems? So to give you a bit of an idea of how we conducted this study, this was done in the UK Biobank cohort, which is it's really an amazing cohort. It was collected in the UK, and it involves both brain MRI, cardiac MRI, abdominal MRI, as well as extensive phenotyping, blood chemistry, urine, as well as behavioural and physical assessments. And so we considered of the order of 1,000 or so phenotypes, and we categorised those phenotypes as they pertained to the function and structure of 7 body systems and 3 brain systems. The 3 brain systems were grey matter, white matter, and functional connectivity, and the organs or body systems that I've listed there on the right-hand side of the slide there. So, for example, one of the systems was the musculoskeletal system, and the phenotypes that were linked to that were muscle strength and bone mineral density and so on. This is only a selection of the phenotypes. As I said, we had over 1,000 phenotypes altogether. I'm just showing some examples for each of the systems that we considered. Then what we did is we trained a model, a machine learning model, based on the 50,000 individuals that were considered in the UK Biobank. And for each organ or each body system, we trained a separate model to take in that person's phenotypes, take in their chronological age, and learn how to predict that person's chronological age from the organ-specific phenotypes. So this enabled us to train an organ-specific biological clock for the 7 systems, plus the 3 brain phenotypes that I've shown here. So this is the performance of the models that we've trained. What I'm showing here on the x-axis is chronological age. The y-axis is the predicted age from the model, and we can see that we trained a model including all phenotypes. That's what I've referred to as the body, up in the top left corner here, and we achieve quite good accuracy. We can achieve a correlation between our chronological and predicted age of 0.8, 0.79 actually. As expected, when we move to specific organ systems, that correlation drops. So the performance of the model drops. We can't predict the person's chronological age as well as what we do for the whole body, but we can still achieve relatively good accuracy. The worst performance was for the immune system, where accuracy drops to a correlation of 0.3. So just to summarize once again what we've got organ-specific phenotypes, train a model, predict a person's chronological age from that, and the gap between their predicted age and their chronological age gives us an indication of whether that organ appears younger or older than expected relative to peers of the same age and sex. Similarly, we did this for the brain, and we were once again able to achieve quite good accuracy when we integrated all 3 brain phenotypes. The accuracy dropped somewhat when looking at specifically grey matter or white matter or functional connectivity. So we're excited about this because this really gives us a personalised multi-organ age profile where we can, as I've shown here for individual 1, we can characterise whether each of the 7 organ systems appears younger or older than expected. And we think that this can be used not only as an early diagnostic tool, but also to understand relations, as I'll show you in a moment, between organ systems. The other thing to note here is that most of the phenotypes that we're looking at here are routinely used in the management of clinical management of older individuals. So in terms of translating this work into the clinic, it is feasible. I mean, some of the phenotypes, particularly the imaging phenotypes, would be more challenging to acquire en masse, but blood chemistry and physical and behavioural assessments would be straightforward to implement. So the first thing that we wanted to understand is whether there is synchrony between these clocks. If your heart is older than what you expect, does that mean that your brain is also older? And on one level, you can say, well, it's kind of obvious, right? Aging is a whole body thing. If your heart's older, obviously your brain is older. But as I'll show in a moment, there are selective and specific influences where one organ system specifically influences the rate at which another organ system ages. So first of all, we looked at the relationship between organs, between body organs and brain systems. We weren't able to look at the opposite. And the reason for that was a methodological reason, is because brain imaging was performed after body imaging was performed. So making causal inference in the opposite direction is challenging. We only looked at the forward direction. And we see that there are influences from the renal, from metabolic, cardiac and musculoskeletal, but by far the strongest influence is the cardiac system. And so what we see here, the way to interpret that, that weight that's fitted to the link that moves from the heart to the brain, is that each 1 year increase in cardiovascular age leads to approximately a 1 month or 27-day increase in brain age. And I mean, a month might not seem a lot, but this can accumulate over years. And so this shows the selective and specific relationship between heart and brain. Now, as I've mentioned previously, there is this concept of age gap can be difficult to interpret, namely because the gap may have emerged very early on in development. And it doesn't necessarily, if your brain age gap is positive, it doesn't necessarily mean that your brain is aging faster. That gap might have emerged during adolescence or at a particular critical period in your life during illness, but that gap then remains constant from that point on. To make more specific inference that what we're actually looking at here is aging or acceleration in aging, is that we looked at longitudinal data. So in a subset of the individuals that were studied here, we had follow-up measurements, follow-up imaging, as well as follow-up behavioral blood draws and so on. And this enabled us to compute the rate of change in age gap, to really understand whether what we're looking at here is something that happened early on, or actually acceleration or increased aging. And to do that, what we did, we computed in this sub-cohort rates of change, and then we tested whether the age of a certain organ system would causally influence the rate of aging of other organ systems. And this led us to a multi-organ aging network. And this network shows selective and specific influences on how, for example, at the top, we see the pulmonary system. And this shows that the age of the pulmonary system selectively and specifically influences the rate at which the heart ages. And so we can see these characteristic aging pathways in the system. For example, aging of the pulmonary system leads to faster aging of the heart. Faster aging of the heart leads to faster aging of the musculoskeletal and the renal system. Some of the interesting characteristics here are that we have so-called hubs. So the musculoskeletal system is a hub of the network. There are multiple other organs that lead into or that result in faster aging of the musculoskeletal system. We also looked at whether there are certain lifestyle or environmental factors that are associated with either older biological age or younger biological age. These were factors that once again were measured in most individuals participating in the UK Biobank. And the way that this visualization is organized is such that the features on the right, the environmental or lifestyle features on the right, are associated with an older appearing organ system, whereas those on the on the left are associated with older. The ones on the right are associated with younger organ systems. And we stratified it by the organ system, which is shown there in the middle. There are some obvious and well-known effects here that we pick up. So, for example, smoking is, as we very well know, is associated with effects in the pulmonary system and the heart. Deprivation. We also found some more unusual factors. I won't discuss those in detail here. And on the other side of the coin, on younger biological age, we see that factors such as physical activity and access to green space are associated with younger biological age. Now, it's important to note these are not causal. This is a correlational or an association analysis. So we really need to do further work to test whether these effects would in fact be causal. The other important thing to notice is that the effect sizes that we're looking at here are relatively small, particularly on the right-hand side of this plot, where we're seeing that the associations are in the order of a correlation of 0.1. So any of these factors by themselves are probably not going to have a profound impact on aging, but there may be a cumulative effect when combining many of these factors together. We did a similar thing for brain age, so factors that are associated with brain age. I won't go into the detail of that here, though. That's true. We then looked at diseases, and what we wanted to ask is whether there is an effect of organ-specific aging in individuals that had been diagnosed with one of these 16 chronic diseases, the ones that I've shown there on the y-axis. So we stratified individuals according to these 16 diseases. The top there is healthy controls. These were individuals that were not diagnosed at the time of scanning or blood and physical assessments. And the columns here correspond to our organ systems. And so what I'm showing here is the mean age gap across disorders and diseases. Perhaps an easier way to visualize that is here, where each icon, I mean, the x-axis here is the mean age gap for the particular disease and organ, and the icon is shown, is positioned at the gap. So, you know, for example, here we see for CKD, chronic kidney disease, we have a mean age gap of around 2.1 or 2.2 or so. So that means that the kidneys appear, the lungs, the lungs appear 2.2, on average 2.2 years older in chronic kidney disease compared to healthy controls of the same age and sex. There are some obvious things that pop up here. So as I've highlighted, in the case of chronic kidney disease, as we expect, the kidneys appear the oldest. In the case of pulmonary disease, it's the lungs, right? It's what we'd expect. In the case of multiple sclerosis, we see that the brain is the organ that appears the oldest. But there are also some interesting and unusual effects where we see the organ that's not typically associated with primary disease pathology also shows extensive aging. And so one that I've highlighted here is the case of schizophrenia and dementia, where we see profound or significantly older aging of the pulmonary system in schizophrenia and dementia, close to what we see for the brain. And of course, pulmonary system is not typically associated with psychiatric illness. We also see that the brain, aging of the brain is significantly greater across all of the 16 diseases considered in the study, as I've shown there with the yellow line. And this really highlights, at least for us, that there is primary disease pathology, but the effects of aging can be seen across multiple systems, and in the case of the brain, across all of the disorders that we've considered here. I'll quickly run through some other visualizations, how we visualize that. In these clocks, this is once again, we stratified aging either body or brain. Left is body, right is brain, and we've positioned the specific disease along the different points of the clock. This is another one that's stratified by the organ system, where the size of the text is modulated by the disease that shows the greatest age for that organ system. So if we look at the kidney, which is there on the bottom right, it's chronic kidney disease, which shows the greatest age, which we would expect. But once again, there are effects there that require deeper investigation. So, for example, in musculoskeletal system, for the musculoskeletal system, it's cirrhosis that shows the greatest gap. Now, one thing that you might be thinking of, I'm talking about aging here, but how do we know that these effects are not the effects of disease itself? How do we separate the effect of aging from the effect of disease, or are these one and the same? And we did various analyses to try to tease apart these two effects. I won't go into the detail, but what I'm showing in this plot here is on the bottom, on the x-axis, we're using a t-statistic to quantify the effect of the disease. This is effectively done by comparing healthy controls to the particular patient cohort, whereas the y-axis in these plots is the effect of aging as quantified from our machine learning model. We're essentially taking the coefficients from the model and placing them on the y-axis. And we see a relatively strong correlation between the two effects. And so this tells us that it is difficult to differentiate the effect of aging from the effect of disease itself. This is very general. We've done this also for each of the specific diagnoses that I've considered here, and we do see similar associations between the two. So you might be wondering, well, what's the actual practical or clinical utility of having personalized organ-specific age profiles? And I'm going to give you some examples of that to wrap up now. And one of the interesting things that we have available to us in the UKB is survival data. So these individuals were followed up 13 years post the acquisition of brain and body imaging. And of those individuals that were followed up, 8,000 were deceased in that period, in that 13-year period, and 135,000 were still alive. And so we looked at, one of the obvious things to look at was whether the body age gap at the assessment period was significantly different between those individuals that were deceased and no deceased and not deceased. And we found significant differences across all organ systems, profound and significant differences in that body age and brain age gap was greater in the deceased individuals. So this started to think, well, can we actually use the personalized organ age gaps to predict the risk of mortality? So we've trained a Cox proportional hazards regression, which is in statistical terms, it's a mortality prediction model. And what we put into this model is our organ-specific age gaps, as well as well-known factors that are predictors of mortality. So these are the diagnostic status if the person was diagnosed with any of the illnesses that I've listed there on the right, as well as obviously their age and sex, which are strong predictors of mortality. And what we see when we fit this model is that we can do quite a good job of predicting mortality. We achieve an AUC of 0.77, and we see that specifically the overall body clock as well as the pulmonary clock are significant predictors of mortality above and beyond what we get from diagnostic status, age, and sex, and the other factors that I've shown there. So this tells us that there is utility in adding personalized organ-specific aging profiles in predicting mortality above and beyond what we can predict using diagnostic status, age, and sex, particularly the pulmonary system and whole body age. We also ran some supplementary models where we aim to predict survival time. This is 5 and 10 year survival time, and a concept known as premature death. And what we showed in this was once again that while age, sex, and diagnostic information were significant predictors of mortality, as we would expect, adding in organ-specific age gaps improved our prediction performance. So to wrap up the study, we developed for the first time personalized body and brain biological clocks. So this was using not only imaging phenotypes, but blood and behavioral and environmental phenotypes. And this really enabled us to study synchrony among different organ systems. We mapped a multi-organ aging network, and this revealed specific pathways whereby the age of one organ system selectively influences the rate at which another organ system ages. And we see particular characteristics in the network, such as certain hub organs, such as the musculoskeletal system, and pathways via which aging propagates through the system. We also mapped disease-specific organ aging profiles across 16 chronic diseases. I would like to thank the collaborators who have been involved in this work. The work was led by Ye Tian. It was also done in collaboration with Vanessa Cropley, Andrea Ma, Nicola Lautenschlager, and Michael Breakspear. So to wrap up, to conclude, I think the key point that I want to get across is the brain is not an isolated organ, yet neuroimagers and neuroscientists invariably study it in isolation of other body systems. And understanding the brain requires an understanding of how the brain influences and is influenced by other organs and systems. And so I think that a whole body PET will be a key tool that will enable us to take this area forward, the area of network physiology, and truly begin to understand relationships between different organ systems, particularly as they relate to the brain. I also see huge clinical potential here, not only in personalized and organ-specific age profiles, but in using this information to predict illness prediction and also enable early diagnosis. I think, as I've said before, a lot of the phenotypes that we've used in our models are already routinely acquired in the management of older individuals. So taking this to the next step of implementing in a clinic, we hope would not be a big step from where we are now. I'll stop there. Once again, I'll thank Fernando and Steve for inviting me to come up.